Good afternoon, ladies and gentlemen. Welcome back to another ShareWise webinar. Today, we are, presented with the Managing Director, Liz, from Argenica Therapeutics. I'm very, very excited to see the in-depth of the company and their outlook moving forward in the next year, 2 to 3 years. I just want to thank Liz for coming here today and doing a bit of a presentation, a little bit of a Q&A, a little bit more of an interactive webinar with everyone there watching. There will be a very, very good chance for you guys to ask any questions you would like, any answers you're looking for. Well, today's the day, definitely. My name is Harry McCaskill, one of the senior advisors here at ShareWise. But Liz, again, thank you for your time. I'll let you sort of introduce the company a little bit more, introduce yourself, and then we can jump into a few questions and just take it from there.
Thanks, Harry. And my PowerPoint is now working. So I was thinking there's probably quite a few newer shareholders to this story. So it might be worth just diving into just a couple of quick slides on the presentation that I can run through just to introduce the company a little bit more. And then, of course, really happy to take questions. So if that works.
Yeah, that's a great idea. Go through a few key updates and whatnot for those new potential investors, and give them a little bit of insight into what's been happening over at the company.
Okay, so you can see that all good?
Yeah, we've got all eyes on that.
Okay, great. So I'll just quickly sort of introduce, I guess, the company and what we're trying to do. So we did list in June 2021, and the company was formed not that much sort of prior to that, so sort of just at the end of 2019. The drug asset that we're looking at, it's known as ARG-007, and it's spun out of the University of Western Australia and the Neuroscience Research Institute based here in Perth, which is where I am based. And the drug itself, I used to actually sit on the board of the Neuroscience Research Institute, and we on that board, and our Chief Scientific Officer, Professor Bruno Maloney, was starting to generate some really exciting data on this drug asset. And so as a company, we have the benefit of having around a decade of research work that has gone into this drug. So we actually know a huge amount about it. In drug development, what we really do try to do before we take any drug into the clinic is we do try to de-risk the drug asset in a number of ways. So that's using sort of animal models, laboratory studies to really understand a lot about how well the drug is working in a particular disease indication, but also how it's working. So what is the mechanism of action of that drug? How safe is that drug? How well tolerated is that drug? So that has sort of culminated in over 25 peer-reviewed publications on the drug itself, plus a whole raft of other data that we have sort of sitting behind the company. So that, for me, I guess the thing that really drove me to wanting to take on the CEO role in this company from sort of pre-listing was really just the quality of the science that we have to rely on, which I guess gives us a little bit of comfort as we're moving this drug asset into the clinic.
Yeah.
The
science in this sort of area is key, and you've got a little bit of an edge there, so you jumped on it.
Yeah, absolutely. So, I mean, the science is what I love. The other thing about the science is, obviously when we come to start having discussions with large pharma companies to hopefully do a deal on this drug asset, is that it gives them a huge amount of data to do due diligence on as well. So, I have the benefit of being able to hand over reams of data, both sort of published and with what we've got in the company as well. So that makes my life a lot easier, and really allows me to have a conviction around the story as well.
Yeah, I imagine.
So in terms of where the majority of our data sits, it does sit in looking at the efficacy of the drug in acute ischemic stroke. And I might sort of touch on a little bit more around what that actually means and how the drug is working that indication. But just the mechanism of action, the way that the drug actually works, it does lend itself to quite a number of other neurological conditions. And so the main ones we're focusing on, one of the ones that we're really excited about is traumatic brain injury. We're starting to generate some really exciting data in that area, but also a rare pediatric condition known as hypoxic ischemic encephalopathy as well. And I can come to that in a bit. But if I sort of focus on stroke in particular, so what we're really looking at is an ischemic strokes, and these are strokes caused by a clot in a blood vessel in the brain. They make up around 85% of all strokes. The other type of stroke is known as a hemorrhagic stroke, and that's a bleed on the brain. We don't have any data in hemorrhagic stroke, but it is potentially, the drug could potentially work in that indication. But given the biggest market is in acute ischemic stroke, that's the market that we've focused on, or the indication that we've focused on. So essentially what happens when a blood clot is lodged in a blood vessel in the brain, it obviously stops blood flow to that part of the brain, and the brain needs oxygen. Brain cells last about 3 seconds without oxygen, so in that immediate vicinity of where that clot is. And so we know that 1.9 million brain cells die every minute that blood flow is restricted, and essentially that's what's causing the stroke. And what we see that happens in the brain following a stroke is you get this sort of cascading out, or this proliferation out of the brain injury. And that's because once one brain cell has an oxygen deprivation, you get calcium that rushes into that cell, and it causes that cell to sort of implode. And it just triggers this whole sort of cascading out of brain injury. So what our drug does, it actually blocks that calcium influx into the brain cells. So it sort of hibernates those brain cells and stops that cascading out, or that proliferation out of brain injury. And that calcium influx, that's not only seen in stroke, we actually see it in traumatic brain injury. We see it in neurodegenerative diseases, Alzheimer's, Parkinson's disease. It's called exocytotoxicity. That's the actual sort of scientific name for it. And that process is actually seen in a number of neurological conditions. So the fact that our drug stops that process, for us is really exciting, because it does open up sort of a world of opportunity. But I do often like to say to shareholders, as a CEO of the company, the worst thing I can do is have a scattergun approach and try and go after every indication. We are extremely focused on really proving up the efficacy of our drug in stroke patients through that clinical trial program of work, whilst looking at what is that next indication that we will potentially bring into the clinic. I guess going into a bit more on the data that we do have. So one of the most compelling preclinical studies we have is in a non-human primate model that pretty much exactly mimics what happens to a human stroke patient from a clinical point of view. So this is where we occlude a large vessel in the brain, essentially blocking that blood flow, which sets off that process of calcium influx and that cascading out of brain injury. And then what happens now in terms of standard of care of stroke patients, that clot can actually now be removed through a procedure called thrombectomy. So we removed that occlusion in this animal model, and then we looked at how well our drug was working in protecting that brain injury. And so what the startling results of this study was that our drug was able to reduce that brain injury by 70%, which is hugely significant. And that even though it's only a single dose that we're delivering, we get that reduction, that 70% reduction is lasting we looked out to 28 days. So it's a permanent reduction. So any reduction, obviously, that we have in brain injury that stays whilst we're looking to restore blood flow is obviously going to have a better functional outcome improvement for these patients. So where we're at now, we've done a number of other studies. We know that our drug doesn't exacerbate bleeding. We know that it can also be given with a standard of care drug that some stroke patients get. This is known as alteplase or TPA. It's a drug some stroke patients are eligible to receive if they arrive at hospital within a certain time frame. And this drug dissolves.
What is that? Sorry, what is that time frame there?
So it's around 4.5 hours. So anything beyond that, the drug doesn't really show much of a benefit. So the patient has to arrive within 4.5 hours. The drug itself, so it dissolves the clot essentially. So if someone has a propensity for bleeding or there's some comorbidities, they also won't be eligible for that drug.
Yeah.
But in any drug development, one of the critical steps is to make sure that your drug has no impact on that standard of care drug, but also that that standard of care drug doesn't impact your drug and potentially change your drug's efficacy as well. So we've published data looking at that as well. And then we've undertaken a phase 1 clinical trial. So this is showing the safety of our drug in healthy people. So volunteers coming into a clinical trial facility here in Perth. We did a dose escalation, so looked at 4 different doses in those healthy volunteer cohorts and saw nothing, which was great. So no serious adverse events reported in that trial. So that really then gave us the confidence to move into the phase 2 trial, which we're currently in at the moment.
Yeah.
So really what we're trying to prove in this trial is first and foremost that the drug is safe. So one of the things I like to sort of really reiterate to people is that in neurology, you have to prove first and foremost, the biggest regulatory hurdle is to show that your drug does no harm.
Yeah.
So it can't exacerbate anything that's going on in stroke. So stroke patients do have a propensity to have some hemorrhage, some bleeding, some changes to intracranial pressure that can result in sort of worse outcomes. So we really need to make sure, obviously, our drug isn't doing anything around that, and it's really doing no harm to these patients. And then a secondary thing that we're looking at then is, well, how well is the drug actually at reducing that total amount of brain injury in these patients? So they're the sort of key ones that we're looking at. But if we show safety, it really is the green light then to move into the next phase of the clinical development, which will be a large phase 3, what we call a confirmatory study.
When are you expecting phase 2 to get the results in phase 2, get those readings, and be able to make that decision and obviously move forward?
Yeah, so at the moment, we are a quarter of the way through dosing of patients in the trial. So we're dosing 92 patients in this trial. And I think I've got some on the next slide, just a bit of data around how that looks. So recruiting or dosing 92 patients in the trial around in 10 emergency departments across Australia. So we're at 20, we've sort of reported we're at 23 patients at the moment. We've got a few more beyond that. They're sort of continuing to come in, but that's one of the key sort of safety milestones that we look at, that sort of first 23 patients.
Yeah.
And so that they were recruited at 5 of the 10 hospitals that have been activated. We now have other hospitals activated. So by the end of this year, I'll be able to give shareholders a really good sense of when we expect that data readout. Yeah, but I imagine it'll be towards the end of next year.
Yeah, because Rob was asking, are you still expecting the results from that the first 23 patients to be released before the end of August?
Yeah, so it'll be early September. So we've now locked in the Data Safety Monitoring Board meeting. Yep. That Data Safety Monitoring Board reviews all the safety data from those first 23 patients. The biggest challenge in that DSMB is actually coordinating calendars. We have 4 independent neurologists from around the world that sit on there. They're all still practicing clinical neurology, so just coordinating calendars. So we found a date now. It'll be early September. So yeah, we will have the results of that out in early September. Right. But it doesn't mean we can't continue recruiting patients whilst we're waiting for that DSMB, which is exactly what we're doing in the trial. So hoping I'll just say, in terms of the recruitment in the trial, the feedback that we're getting from the principal investigator neurologists at each of the hospitals is that it's actually a really easy trial to recruit into because it follows exactly the standard of care that these stroke patients get. it's really just adding another IV infusion drug in the emergency department, which the stroke care nurses are doing all the time. So they're even doing it, they're doing it for that clot dissolving drug I was talking about before. So it's actually a really easy trial to run. The other thing that is good about the trial is that because it's a standard of care follow-up, so if I just pop back to sort of looking at this screen, so we're collecting safety data at day 1, day 2, day 3, day of discharge, day 30, day 90, and we're looking at sort of brain imaging on those time points. We're doing functional assessments, but all that is actually standard of care. So we don't have to pay extra for it. We don't have to pay extra for these MRIs. It is already what the hospitals are doing. So it does actually make it a reasonably cost-effective way to run a trial as well. And given that the neurologists know exactly how to do this because they do this all the time. So so that's good as well.
Yeah, because obviously the cost in terms of the some of the things behind the scenes here can be quite expensive. So that'll keep things level playing field for you.
Yeah, and in particular, like when we go into a phase 3 clinical trial, which will be a big global trial, we'll need to set up more sites. And they can get massively costly if you're having to implement new ways of collecting data or follow-ups or that sort of thing. So given again that it's all standard of care across pretty much every jurisdiction that we will go into.
So how is the company looking and moving towards non-dilutive capital raisings? Are you fast tracking FDA approval, those sort of things, or where's the business standing at the moment with regards to capital and cash flow?
Yeah, so in our most recent 4C, we had 5.9 mil cash in the bank. So we're really well funded from that perspective. So that gets us well and truly past the end of our phase 2 clinical trial, which is great. In terms of non-dilutive funding, we're always looking for non-dilutive funding. So, I did flag to a couple of ministers recently that our nearest competitor, a Spanish company, has managed to get quite a lot of non-dilutive funding. So if they want to fund a good Australian company, then please throw it our way. So we have attracted close to 5 mil of non-dilutive funding anyway, which is fantastic. But always looking for opportunities to get more of that non-dilutive funding.
Of course.
Yeah, in terms of the FDA, so I did in our IND submission. We're just in the process of preparing our IND at the moment. And so what that will allow us to do is once we get an open IND with the FDA, we can apply for regulatory sort of, I guess, incentives type of thing. So I've got there the fast track application. So this allows a sponsor of a clinical trial in the US to get the review of their sort of regulatory documents fast-tracked, where you're addressing a large unmet medical need, which stroke obviously is one. We'll also look at things like breakthrough designation and things like that. So other things we can look at from a regulatory incentive point of view to
And then I guess for the company, are you is there a likely application beyond the stroke side of things? And obviously, will that sort of lead to more phase 1, phase 2 sort of trials?
So we're actually I can flick through a couple of slides here, but in terms of our TBI data, so we've got the rat and the ferret data that we did pilot studies in. So even pre Argenica in Bruno Maloney's lab, he had collected some really positive data on traumatic brain injury. This is really looking at that kind of moderate to severe TBI, which would be akin to like a king hit, sort of like domestic violence. We see it often, or motor vehicle accident, or like a severe concussion on the sporting field where you're pretty much knocked out type of
just major brain trauma, right?
Yeah, exactly. So really what we're looking at here, so when someone sustains a TBI, you get what's known as diffuse axonal injury. So this is injury that actually sort of, even if the knock is on the top of the head, you get this injury that sort of diffuses in the brain, and we see an accumulation of proteins in a key area known as the hippocampus, and I've sort of pointed it out there on that slide. And the reason that the hippocampus is really an area of interest is because this is where it's responsible for memory, essentially. So when we start seeing things like dementia in people that have suffered a number of TBIs or concussions later in life, it's because we start getting the aggregation of these proteins. So amyloid precursor protein, obviously the amyloid protein is what's implicated in neurodegenerative diseases, in particular Alzheimer's. We also get another key protein, neurofilament proteins, that start to aggregate. So in these studies, we're really interested in this area of the brain, the hippocampus, and how well our drug actually reduces the aggregation of these proteins. And so in both the rat and the ferret pilot studies, we saw that our drug was having a pretty significant ability to reduce the aggregation of these proteins in this key area, the hippocampus. So that's then given us the confidence to do larger studies in this. So we've got ongoing rat and ferret studies, and we will have data readouts from those studies in the next sort of 6 months and 12 months. So that's really exciting. And then we are looking at doing more of a mild type of concussion study as well, just to confirm in that where we're having, you know, a repeated concussion. So it's not sort of like a one-off, but where you might sustain a concussion multiple times, how well the drug sort of works in that respect.
So the business is focusing on the stroke drug initially.
Yep.
Once it gets up and running, well, then more time will be spent into the TBI and things like that, just to continue growing, right?
Yeah, exactly. So with the TBI, so the moderate to severe, these patients, they'll come into emergency departments. So we'll be able to run a trial really similar to the stroke study. This is a single acute dose IV infusion application of our drug in this more severe TBI. So given we've already done a phase 1 with this drug, the dosing looks pretty much the same in terms of patient's dose. We can go straight into a phase 2 clinical trial in emergency departments for TBI. So that's another key benefit. So putting, I guess, another string to our bow in terms of neurological indications would be really compelling.
Yeah, and these things certainly don't take a couple of days. It's definitely it's a slow grind and a slow build, that's for sure. We've got a question, Rob. He's asked, who is the best listed company to compare AGN to, and what was the major catalyst for this company that provided their share price with a re-rate? Asking who are your main competitors, and where do you see yourself compared to those sort of businesses on the ASX?
Yeah, so I guess we sort of look at some comps around sort of level of or stage of clinical development or neurology, I guess. In terms of the most similar, Neuron probably is the most similar. It's a peptide drug. They had previously done some work in TBI. Their drug works on inflammation, so it's sort of slightly different mechanism in terms of our drug. And they've gone into that sort of rare pediatric indication, which we do in HIE. We also have that. So typically, though, where you see, re-rates and maybe actinogens, an example of where you would see, I guess, people getting that sort of looking at that value inflection around a phase 2 readout. Obviously, you just hope the data goes the right way. So what we keep saying is we've got so much preclinical data. We pretty much can't de-risk this drug anymore in terms of the data that we have. There is always obviously a risk when you go into humans. But the models that we're using in the animals really just so closely reflect what's happening in the human clinical setting as well. So so we're hoping, and it is the phase 2. It's safety. It is the obviously the key thing to get us to the next stage.
Yeah, it's all about minimizing the risk, right? Like making sure it's as safe as possible, so you don't have any of these patients that are being given dosages to have a bad reaction or get one of those bad results, which is going to significantly hurt the you know, the business or the reputation and really throw a bit of a spanner in the works. So it's the old saying, it's better safe than sorry, that's for sure.
Yeah. And it's, I mean, in neurology where you've got, you know, so neuroprotection, the ability to protect brain cells following stroke, there's no drug on the market, right? So the efficacy bar is, you know, can you do better than nothing, right? So the safety bar is huge. Like you cannot do any harm with your drug. The efficacy bar, we've sort of seen it with Alzheimer's drugs, where I guess the efficacy is reasonably questionable, but the general view from the FDA is show us that, you know, there's, you know, you've got a bit of an improvement in these drugs. Let us get them on the market, and then we can see sort of how well they're doing. As long as they're not hurting anyone, there's no harm. Let's actually get them out to these patients.
Yeah, for sure. We've just got one coming from Pete. Do you have a vision on final outcome for Argenica? To sell to a big pharma or license model like Neuron?
Yeah, so we you know, and I guess I speak to, you know, I've spoken to John about this at Neuron, I've spoken to Nina at Dimerix around, you know, that sort of partnership model, and typically a pharma company wants to see the phase 2 data, right? So you either typically get deals in preclinical where they're buying in a cheaper asset but taking on more of the risk, or they'll do something post phase 2 where they're de-risked it in sort of a clinical trial, but they're going to have to pay a lot more, obviously, for the asset. So now where we're at is that kind of that phase 2 readout. We have had a lot of engagement with pharma companies. We're under CDA with a couple of them. As I said before, we've got a huge amount of data that they can start doing due diligence on. So, but really what they want to see is that phase 2 data to give them comfort that, you know, the asset, I guess, is not only safe, but they're showing at least a trend to efficacy as well. But really, I guess the strategy of the company, so a phase 3 trial isn't going to be hugely expensive. You know, it's not going to be, you know, hundreds of millions of dollars. So, you know, we're sort of paralleling the strategy around really pushing around that pharma partnering, but also looking at, you know, we can go it alone. If we need to do a phase 3, we can go it alone. And once we get that IND, really confirming with the FDA that phase 3 clinical trial is sufficient to be our registrational trial. So the only trial that we need to do to get that drug on the market. So we could potentially go it alone, and we could do, I guess, like a Dimerix has done, and do a deal sort of in the middle of that phase 3 as well. So sort of really trying to parallel both those strategies, so we're not delaying actually being able to get it into a phase 3.
Yeah, okay, that makes sense. Well, Pete just said thank you for the update today. So
no problem.
You've answered his question very, very well. Are there any other things that you've got in the pipeline that, you know, big catalyst or key little milestones that investors can expect to see over the next 6 to 12 months that you sort of haven't touched base on too much already?
Yeah, so it's really, I guess, sort of keeping the market updated on how recruitment is going. We'll obviously have the DSMB readout in early September, continuing to keep the market updated on recruitment numbers, because that'll give the market really good sense of how well the recruitment's going and when we're likely, obviously, to get a readout. So that's on the trial, really looking at the data in the TBI, so having some of those preclinical data readouts as well. We've got more data coming out on HIE as well, and hopefully some Alzheimer's data coming out as well. So Alzheimer's is probably, I mean, it's really hard indication. We've got some exciting data in that, but it's, as I said before, we're sort of prioritizing where we've got, you know, more breadth of data, so really that TBI and that stroke area as well.
Yeah, for sure. And then with I guess moving forward for the shareholders that are either in now or looking to get in, what's the experience with everyone at the firm, and how many people do you have working behind the scenes trying to get things done?
Yeah, so we obviously, my background, I do have a PhD in neuroscience, so I do sometimes have to, you know, roll my sleeves up on the science side of things, but I do actually love that. But we do, so we have a vice president of clinical development who is running the trial along with Alithia. So we have a person dedicated to sort of helping us run that trial as well as obviously our global CRO running that trial. We have also a lab based at Perrin that our Chief Scientific Officer runs, so a number of RAs. So that's sort of churning out the preclinical data. We have our postdoctoral research fellow, Dr. Adam Edwards, who's running all our HIE studies as well. And we have just recruited someone into the team who I'm so excited about, but I'll announce that to the market when they come on board. So he has an amazing pedigree. So he'll come into the team really to look at, pushing that sort of next indication into the clinic. Yeah.
That's very
the other area, sorry, I'll just quickly say, because we're really trying to look at the potential of this nasal delivery as well of our drug in those sort of chronic conditions such as concussion, etc. Yeah.
And then with, I guess last thing from my end, and obviously probably a few other people probably a little bit curious on as well, is how easy, is there a limit to the amount of patients and recruitments you can make for these trials? For example, the phase 2, you think you said 92 or 93 at the moment. What's the ideal number and how realistic is it to be able to get there?
Yeah, so the 92 number really comes from the biostatisticians that we had working with us on that protocol. So really we came up with 92 as a way to look at how many patients would we actually need to sort of show the safety. So that's powering it sort of for safety, but also to see a likely trend or signal in the efficacy as well. So we spent a lot of time looking at all the preclinical data, but also mapping that onto a phase 3 trial that another company had done to see sort of where they were seeing their signals. So a lot of work went behind that 92. I know it sounds like a bit of a random number. When you put in your ethics application, that's what you go for. So we can't recruit more than 92. That's the number.
Right.
So coming to a phase 3, what we then do is we power it for efficacy in a phase 3. So we really are looking at, how well this drug's working. So we'll take all the data from the phase 2, get the biostatisticians to work their magic. That is an area of science that really just bamboozles me. They're extremely intelligent individuals. So but they will work out, well, how many people do we actually need in that phase 3 trial?
Right. So it is, okay, that makes sense.
Yeah. And what I'll say, in terms of ease of recruiting, so the feedback we're getting, this protocol is super easy to recruit into. And then there's unfortunately, sadly, there's no shortage of stroke patients.
Yeah.
So with the phase 3 trial, the more sites we bring on board, the faster the trial will go. Yeah. It is a really attractive trial in that it's short. So the 90-day follow-up, it's a single dose. It's the patients come to us. We don't have to go out and promote the trial. They're arriving in emergency departments. The consent's easy. So in terms of recruitment, I'm super comfortable that it's that's very doable.
Excellent. And within those 90 days, obviously, the patients they're taking one dose. Are there any external factors that are that do significantly impact the results of the dose and what the drug's actually doing? For example, like their diet or the amount of time they're spending on certain activities or whatever. Is that easy to track, and does it significantly skew the results?
So that is such a fantastic question, and something that I think the FDA really grapples with neurology, because ultimately, as drug developers and as regulators, what we want to see is that the quality of life of these patients changes, right? So we're looking at, in the phase 3 study, we're looking at a sort of functional and neurological outcome assessment to really understand, has that patient actually shifted on a predetermined scale? In neurology, that's hard, right? So patients typically get discharged from a stroke around day 6. Yep. They go home. Some of them will do physio every day, right? And we know there's a level of neuroplasticity in the brain.
Of course.
So what the FDA now is kind of really starting to think about is biomarkers. So what are those more quantitative measures like brain imaging that are going to be less impacted by some of these external factors, such as, you know, rehab up to 90 days. So,
yeah, and I would
really good question. Yeah.
And I would imagine someone who's eating a healthy diet and then getting outside more so than someone who is obviously being impacted by the stroke, who's just at home, probably not eating as well as they should, or not getting out of that. It's probably going to impact things. So that's obviously something that you're grappling with on your end as well.
Yeah. And that's why we'll continue to collect that brain imaging, that reduction in brain injury throughout the phase 3 trial as well. Yeah.
Excellent. Well, I just want to say thank you for coming here today, Liz.
Thanks, Harry.
A little bit of a Q&A, a little bit of an interactive one today, and I think there's been a lot of value there for shareholders and anyone looking to learn a little bit more about the company. Is there any last word you want to put out there before we call it a day?
Oh, look, just, I guess, within us as a company, we're really excited. We're just so pleased with how the clinical trial is going, and just the engagement that we've had with the neurologists and the sites have been absolutely amazing. And we just, it's just such a privilege to work with these people and, really hopefully change the lives of stroke patients. Every 19 minutes in Australia, someone has a stroke. So 1 in 4 of us will have one. So it's a huge unmet need. We're just really excited to see the data and how it all goes.
Yeah, excellent. I mean, the technology, it's only going to get better, and hopefully, the impacts of the stroke just get less and less over the years.
Exactly. Yeah.
Well, Liz, again, thank you for your time. I know you're a busy lady, and everyone watching, thank you again for coming and watching another ShareWise webinar. There will be many more to come. But if anyone has any queries, feel free to reach out with us here at ShareWise. You can go to our website. There's a speak to an advisor form you can fill out, and we're more than happy to have the conversation. Liz would always, I would imagine, be always happy to have a conversation or respond to emails or whatever if she can find the time. So it is recorded. It will be sent to Liz. She'll have access to that and she'll send it out. Also, it's on our YouTube channel. So again, it's a pleasure. And Liz, have a lovely, lovely afternoon. Everyone else out there, have a lovely weekend.
Thanks, Harry.
Thanks, Liz.